All right, good morning. So let's just start up with summarizing some of yesterday's assignments. So first off, the last bit we did yesterday was trying to submit the job to the MetaPipe functional analysis. So you remember the web portal and everything. So we had two minor problems. Basically, some of you, we didn't know that Finland was associated with Elixir somehow. So some of you logged in with the University of Finland and then you didn't get to log into MetaPipe. So how many didn't get to submit anything because of that? Two? Okay. Yeah, so we didn't know about that. That was unfortunate. Also, I think two of you didn't submit a file to MetaPipe somehow. You probably don't know about it because it's not, you can't see it in the web UI. But all the other jobs ran and finished and everything worked, so that's fine. If you didn't get to submit your files, we already have pre-processed files, so that's not a problem. So that's a quick summary of that bit. We also did some taxonomic classification yesterday, so we followed basically two workflows from the trimmed reads. So one of them we used 16S RNA sequences, this one. So basically with rRNA selector we predicted RNA with HMM profiles. We compared them to a manually curated and small Silva database. And then we did taxonomic profiling using a tool called LCA classifier and visualized that with Krona. We also did the same workflow on clade-specific markers using Metaflam tool and also visualized this with Krona. And you could probably see that there were some differences between these taxonomic classification just based on the databases and the actual methodology you used. So today we are going to do some comparisons. So basically we're doing a similar thing, but we're using a small and simple tool we made ourselves, which basically uses the same Krona tool to compare samples. So we're comparing methods, so basically Metaflam and 16S. We're also comparing some of the samples. So the sample you've got is from baby feces, and they've gotten some different treatments, which is in the assignment. So you should see some differences in taxonomy based on what kind of treatments these babies have gotten when they was born. And we are also going to use a program called Megan to compare multiple samples. So it's this, it's a nice program with a user interface where you can do all sorts of things. So there's a really extensive manual. It will probably take some time, but it's a really good tool to actually visualize some of these things. Yeah, as I said, so Krona Compare, the tool we made, it's basically, so instead of just coloring the chart randomly or after the hierarchy, it colors it after, so basically green means that the fraction is similar to the other data set. So in this example, the bacteria fraction will be pretty much the same as in the other data set, while the red ones, for example, Bifidobacterium, won't be 57%, it will be something else. So it's sort of just a quick tool to get a quick overview of what kind of taxa have similar fractions in the different data sets. So you can basically shift samples up here and browse through them in the same way as you did yesterday, only comparing. Here's another sample. You can see that this part is not as red as this, so probably Clostridiales is close to 48% in the other sample, but not exactly 48. You'll get the grip of it once you start using it. This is a short overview of Megan, so you can do lots of stuff. Here we are comparing 6 different samples with a bar chart view and with a heat map, so you can color it according to similarities and dissimilarity. It's also a program you can just download to your laptop basically. So this program should be installed right in the workstations. So as you notice, the VM is quite small, so you can't really look or visualize stuff on the VMs very good. So both Artemis and Megan will use either, you could use it on your workstations or just download it to your laptop. You can decide yourself.